we as a lineage have been impacting the way things around us evolve as if we are our own evolutionary force. We have become, as we have existed, something outside of evolution. And I think we are essentially a distinct lineage. And what is it about it? Is there something in our DNA that made us so? Probably. Is it one thing? Probably not. Is it a whole bunch of stuff and we're never going to really figure it out? Maybe. But is it cool to think about? Absolutely.
Welcome. How are you?
I'm all right. Yourself?
Good, thank you. So I'm going to just jump in and ask you what brought you and brings you to the work. And for our audience, just to give us a little backstory about how you found your way to do what you do and what you do.
So I'm a scientist. I'm a professor at UC Santa Cruz. I've been here since 2012. Before that, I was at Penn State. And before that, I was at Oxford in the UK. And I work in a field called ancient DNA. And when I got started in 99, it was a pretty new field, but it's not so new anymore. We, as a field, go out and get DNA from things that used to be alive, whether that's plants or animals. I do a lot of work with bones, where you go up into the Arctic and where they're doing active gold mining, or where the spring melt flushes a whole load of water down a stream. It'll peel the permafrost, the frozen dirt off the sides of the riverbeds, or just, on purpose if you're a miner. And along with the dirt comes tens of thousands of bones from things that lived during the last ice age. And it was discovered maybe a couple of decades ago that those things often contain DNA. It's not in good shape, but we can get the DNA out of these remains and we can piece it together. And that can help us to paint a picture of what the world used to look like. And we can ask questions about how populations grow and shrink, when there are local extinctions and replacements, how communities are impacted by things like rapid warming after the last ice age, or the first introduction of humans into a landscape. And we did all kinds of cool stuff through the years. We worked on bison and horses and mammoths, and we're always excited to talk to people from the media about how what we'd learned could be applied to making more informed decisions about how to protect species and ecosystems in the present. But the only question that they ever really wanted me to answer was, how close does this mean, whatever my latest research was, to how we are to bringing a mammoth back to life? And so I got so bored of answering that question. And there really isn't a concise answer to that question, that I wrote a book about it. So I wrote a book called How to Clone a Mammoth that was published in 2015. And that was a lot of fun.
That probably brought you a lot more questions about it than answers.
Yeah, but then I could say, okay, have you seen the book? Right? Because it's in chapter 3. Right. Which I really wasn't able to do before, but that was fun. Yeah. And it started this whole conversation and a whole new way that I've been thinking about work, which is how can we use these technologies that we would need, which I'm happy to get into, to bring a mammoth back to life, to help protect species today. And so that brought me to my second book, which just was published in October, called Life as We Made It, where I really dive a little bit more deeply into how people have been changing things, plants and animals, for as long as we've been people, and then what the new technology that we have today and that are on the horizon might mean in terms of protecting diversity and really trying to make more with the resources that we have available to us today.
So when we say we want to bring back a mammoth, don't you have to start with which mammoth? I mean, because mammoths were around for a long time, or take carrier pigeons or any other species to know which time period we're talking about. Can you actually decide which one you want to bring back, which year of all these, and place?
That's a great question. And it is not a question that I often get asked, but it is the right question. Yes, mammoths were around for millions of years. So what do people want? In general, I think people don't care, as long as it's a big, hairy elephant-like thing and it looks like a mammoth, which is lucky, because we are never going to bring anything back that is identical to any particular mammoth. And really what you're talking about doing is taking an elephant, probably an Asian elephant, because they're the closest living relative of mammoths, and engineering that elephant's genome so that it contains some genes that evolved along that lineage to mammoths. Of course, mammoths persisted till around 3,000 years ago in places like Wrangel Island. So they haven't been gone for that long, but they are gone.
Also, when we talk about the creature itself, do we know what it ate? Can we bring that back? Do we know what it's?
I thought we didn't want to talk about the mammoth.
Well, we're not talking about the mammoth. We're not talking about the mammoths. We're talking about in general, when you bring an animal back. We don't know what it ate. We don't know how
Right,
it lived,
okay.
what it was.
I am
How
totally
how do we
happy to talk about
replicate
the
its-
mammoth. It
Well,
doesn't
it's like, how
matter.
do we say a carrier pigeon, which there were, billions and billions of, but aren't around
Passenger
anymore.
pigeons. These are passenger pigeons. Yeah.
Oh, wait, what's the difference between a passenger and a carrier?
A carrier pigeon is normally a term that's used to talk about like regular pigeons that we have today, but that used to carry the mail around. So people often confuse them because they seem like they're an extinct thing, but carrier pigeons are just normal pigeons. And passenger pigeons are this bird that flocked in the billions, literally billions, until they went extinct in 1914. So they were around much longer than mammoths were. And they are also another species that people like to think about if they want to bring extinct species back to life. But there's a whole different host of technical challenges associated with bringing birds back to life than are associated with bringing a mammal back to life. The most important being that, part of anything that has to do with bringing mammals back is cloning something, like Dolly the sheep was cloned. That's a process called somatic cell nuclear transfer. So this is a necessary component of this. If you're George Church working at Harvard and at this new company, Colossal, and you want to take an elephant and turn it into a mammoth, you have to have elephant cells that you grow in a little dish in your lab, and you tweak them using the tools of gene editing a little bit at a time. Then you have to transform that cell into an animal. And you do that using this process called cloning. We can't do that with birds. Science doesn't know how to do that with birds yet.
But
So
why? Why can't we do it?
it's a hard question. It has to do with the intricacies of their reproductive system. So Dolly the sheep, for example, right? So in order to clone Dolly, they took cells from mammary tissue from the individual that Dolly was a clone of, and then they took from a different sheep an egg cell that was ripe, that was sort of ready to be fertilized. And they sucked the nuclear material out of that ripe egg cell and injected in its place the nuclear material, the DNA that tells it how to be a sheep, from the little cell that they'd harvested from the animal that Dolly was cloned from. In a bird, the egg doesn't become ripe like this at a time that it can be harvested and used in the same way. When a chicken egg is laid, there's a 24-hour-old developing embryo inside that egg, right? And at that point, you can't suck it out and stick something back in. So you need a whole different suite of technologies to be able to do this. And people are working on it. There's several groups around the world who are trying to come up with ways to do this, principally because it would be great to have some mechanism to do captive breeding and reproductive engineering of birds for the purposes of conserving them. Some of the most endangered species on the planet are birds. And so it'd be great to have these technologies, and there are people working on them. We just don't have them. But back to your question, Jesse, about the other things that make a mammoth, or that make a person, or that make any individual the thing that it is. Yeah, I mean, this is a huge challenge. We are more than the sequence of the A's and C's and G's and T's, the letters that make up our DNA code, than the nuclei of our genomes, right? For example, we all know clones, right? Identical twins are genetic clones, and they are different from each other. You can't just say, oh, one is they can just be, swapped in and out without any problem, because they're different individuals. They've had different experiences, and those start prenatally. They have different exposures to different pathogens. They get sick at different times. They have different stressors, and it causes their genes to express differently. They also have different gut microbiomes and different diets, and all of these things are going to influence the way that they look and they act and they develop. I mean, identical twins, as they get older and older, they even look increasingly different from each other, even though they are identical. I mean, DNA really tells us what we look like when we're born. We can tell that by looking at identical twins. Do we know this for a mammoth? Well, we can make some educated guesses about what they ate, right? There are mammoth mummies that have gut stuff in them that we can take out, and we can sequence the DNA from that, and we can learn about microbes, and we can learn about plants, and we can learn about stuff. But does that mean that we can accurately put together what their diets and their gut flora looked like? Yeah, probably not. That the habitat that they lived in is gone. And that's just one of the many reasons that I always say we might get back an Asian elephant that has some genes that have evolved in a mammoth lineage that are able to survive in the cold, but we're not going to get a mammoth.
Can I ask you a question that it's probably not a popular question to ask you, or Jesse won't like? Some species maybe go extinct because it's an evolutionary process, right? I mean, we don't have dinosaurs that we know of. Maybe we do, but
we do. Birds are dinosaurs.
Okay. Oh, there you go. See, this just shows how much I know about all of this. I want to get right into the ethics question. Like, what should we be doing, and what is the value of bringing back the pigeon, the passenger pigeon. What do we learn from passenger pigeon? And how does it help the fucked up world that we live in today? Excuse my French. How can it help us?
There are different people who will make different types of arguments about why you should bring these extinct species back. First, let me address the question about extinction being an evolutionary process. Totally true. Like most species that were ever alive on our planet ever are extinct today, because that's just what happens. And there is a regular rate, background rate of extinction that happens throughout geological time. It's just that there are several specific times in Earth's geological history where the rate of extinction is way higher than that background rate of extinction. These are mass extinction events. So this is really what's happening today. Yes, species are becoming extinct, but the rate of extinction today is many times higher than the background rate in the geological record. And this is why people are arguing that we need to do something about the extinction crisis that we're in. Does bringing extinct species that are already extinct back actually do this? To me, I am much more comfortable saying that these tools that one would need to bring something that is extinct back to life would be far better applied to helping species that are alive today be able to adapt as quickly as possible to the pressures that they're under because of things that we've done to the planet. There are, of course, people who would argue that bringing extinct species back would be helpful. The passenger pigeon, for example. Ben Novak, who's the chief scientist at Revive and Restore, he is supremely fascinated with the idea of bringing the passenger pigeon back to life. And his argument in favor of this is that the passenger pigeon was a keystone species in the eastern forests, and it played a role, a really important ecological role that is missing today. And that is when we had these massive old growth forests, they would come in and they would nest 500 birds to a single tree all at the same time. And they would shit a lot on the ground and they would kill the trees and kill everything beneath it. It was the avian version of a really terrible storm coming through and destroying everything in its path. And we don't have that anymore. And so what is out there that can reset these old growth forests? And if we really are thinking about reestablishing old growth forests rather than having all of these second growth manicured forests, then shouldn't we have back the animal that was actually causing this resetting this process and allowing the ecosystems to evolve naturally? So that's what he would argue is a good reason for bringing the passenger pigeon back to life.
Although it's so hard to really understand the ecology of a forest and what went where. We've done talks with people where it's like, well, these birds are migrating from South America all the way to North America, and nobody has any idea what's going on or how it was affected. Here's a question for you though. Is there any DNA around from before other extinction events where you can look? When we say ancient DNA, how far back can we go in non-living things to find DNA?
Yeah, the oldest DNA so far that's been sequenced is about 1 million years old. And this is from Mammoths. It was a project that was led by Love Dahlen from Sweden. I was a collaborator on this project and it was published this year. Before that, the oldest DNA that had been sequenced was about 750,000 years old, and it was from horses that lived in North American Arctic. DNA that is older than around 50, 60,000 years is rare. So as soon as an organism dies, the DNA starts to get broken down by enzymes that are circulating in their body and in their cells, by UV radiation from the sun, by freezing and thawing that actually, the water molecules expand and it breaks the DNA. And then microbial processes, things just chewing it up from the soil. When we get DNA from a mammoth bone that's 50,000 years old, the fragments of DNA that we can recover are damaged. They have, normally you get these, if I were to do a cheek swab, I could, extract strands of DNA that are millions, maybe hundreds of millions of letters long. But the average DNA fragment that we can get out of an ancient DNA thing is normally 50 letters, if we're lucky, maybe shorter than that. So it decays really quickly. So 1 million years, we can't, no dinosaurs, sorry.
So a stupid question then, does it decay overall? Do you get a bunch of different strands of DNA and some of it is, like you're only getting 50 letters, but is it, you're getting 10,000 50 letter things that are different letters and you don't know how they go together? Like, how do you find it?
Yeah, so it just breaks randomly, mostly randomly. But if I were to extract DNA from a mammoth bone, I get all these different things. I just sequence all of it, and I'll get, billions potentially of 50 base pair strands that I can then take and on a computer, one at a time, try to see where it best matches the elephant genome sequence. And in this way, we start lining them up. It's like a big puzzle piece, and you end up with, I've covered this particular place in the genome with 100 different 50 base pair letters. So I'm pretty sure what the letters there are, and I can do that all the way down here. It's just muddy. If I grind up a whole mammoth, I could sequence its genome billions of times in 50 base pair fragments, line up against an elephant genome and tell you what that sequence looked like. And we do have about a dozen mammoth genomes that we know really well because of processes like this. So we do know what the differences between an elephant and a mammoth were, the genetic differences.
When we see every few years, they find another version of man from going back, and then we say, well, these sequences are different, and this is what this group, Cro-Magnon man gave to us, or these other things. How do you take all that information and then, make a distinction about what it means to what we are now, what it means to be human?
Yeah, that's a great question. So the Neanderthal Denisovan sequences, these are the human cousin lineages of humans that we have a lot of genetic information for now. And by looking at the differences in their genomes to the differences in early human genomes and the hundreds of thousands of human genomes that have been sequenced and published today, you can line them up, and you can use tools of population genetics and different statistics to ask, how long ago did these two genes share a common ancestor? Or, as the team from Leipzig did when they first discovered that there had been some hanky-panky going on between Neanderthals and humans way after the populations diverged. What they saw was that there's DNA from Neanderthals that's in a whole bunch of people that came to them because of where it is in the genome and how diverged it is from other humans after humans became a separate lineage from Neanderthals. And so then you can just make these lists. So where in the genome? So here's a cool fact about this that a lot of people don't know. Most people know that everybody has somewhere between 2 and 5% of their genome is derived from ancestry that is from Neanderthals and Denisovans. Less commonly known is that it's a different 2 to 5% in everybody. And that if we can sum up all of the pieces of Neanderthal DNA that are alive today in humans around the world, we can put together about 93% of the Neanderthal genome. It's still alive. So what does that mean about something going extinct? 93% of the Neanderthal genome is present in us today. But what about that other 7%? And this is, I think, a really super interesting question. Some of that other 7%, where no single person who is alive today has the Neanderthal version, probably just disappeared.
Wait, how do you know that? How do you know that?
Because we have hundreds of thousands of human genomes that have been sequenced, and you could just look for the Neanderthal DNA in these genomes. It's actually that part of it is scientifically pretty well worked out. Like if you go and get 23andMe, it'll tell you how much Neanderthal DNA you have in your genome, right? You can have competition with your family to see who's more Neanderthal-like. It's my sister. She's the most.
Now, when we start talking about the DNA, when we say it's sequenced and we understand it, there's still great mysteries in the DNA that we don't understand. So what do you make of all that?
So this is what I was getting to, this 7%, right? So some of that 7% of the genome is just lost by chance, right? But some of it was lost because if you were a human living in a human family, in a human world, and you had the Neanderthal version of the DNA, you didn't survive, which means that is part of the genome that is uniquely ours and really important for us to be human rather than being Neanderthal. And this is, I think, a really cool area of research that ancient DNA is allowing, just highlighting which parts of the genome those are. We don't have any idea what most of it means, but we now know where to look, and we can start designing experiments to figure out, what's going on there. We had some colleagues from University of California, San Diego, Alison Moultrie and his group, and they picked one of these genes, which is called NOVA1. It's a gene that makes proteins look different during the early stages of neural development. And they discovered by editing the Neanderthal version of this DNA into human cells and then growing those cells into little baby human brains, that the little baby brains, the organoids growing in dishes in labs that had the Neanderthal version looked way different and made way different proteins than the human version. This is one of those places in the genome where no living human has the Neanderthal version. And so this is probably something that is important in making human brains look like human brains, but we don't know how or why. But here is a great line of research for somebody to get into for a couple of careers to figure out what that means.
For somebody watching this, what is the difference between Neanderthal? I love the way you say it. It's very European, Neanderthal, and the human. What is it? Is it conscious? Because we don't know what is the thing in the brain.
It's hard to describe. They looked lumpier. They grew more slowly. They made different types of proteins. That was what was most important. So we were measuring the proteins that they were making and sequencing the transcripts of the proteins that were being made, because this is kind of what's important, we think, with what's going on with this particular gene. But it's so poorly understood that it's really hard to say. But physically, they look different. They were smoother on the outside, actually, the version that had the Neanderthal version, and the cells moved more slowly and the organoids grew more slowly.
Can this help, let's say, in Alzheimer's? In other words, how does this help the world that we live in? Any of this, I'm just trying to understand. I think one of the most fundamental things that we are interested in as people is what makes us unique. Why are we different? Why are we here? Why do we think the way we do? Why do we communicate the way we do and no other species has done this? And I think just it's a fundamental interest to us to understand what it is that makes us human, what it is that makes us different. And sure, as we learn about what it is that makes humans different, we're probably going to learn things that have applications directly to human health, but we don't start with that. You start with the basic science and the foundational insights that then turn into something that's translatable. There's a lot in science that we can learn without specifically trying to save people's lives.
It's also possible we'll learn that there isn't much that's different.
It's possible, true, but we know that that's not true, right?
Well,
so
there's Mozart, there's mathematics, but if we go beyond those things, that level of understanding, planning for the future, those kinds of things, do we think that there's something in the genome that makes that possible? There's some evolutionary trait that's brought that forward?
I think there has to be. Neanderthals and Denisovans, they had stone tools, but we make 787s. And think of the impact. We're talking about bringing an extinct species back to life. What other species is even close to having the capacity to do that? We, as a lineage, have been impacting the way things around us evolve as if we are our own evolutionary force. We have become, as we have existed, something outside of evolution. And I think we are essentially a distinct lineage. And what is it about it? Is there something in our DNA that made us so? Probably. Is it one thing? Probably not. Is it a whole bunch of stuff and we're never going to really figure it out? Maybe. But is it cool to think about? Absolutely.
Do you think that we're actually outside of the evolutionary forces, or that evolution continues on around us and we're oblivious to it?
I think there's probably a little of both. When we took gray wolves and turned them over 30,000 years or however long it has been into Chihuahuas and Great Danes, we were working outside of the rules of natural selection, but we were also capitalizing on them, right? We were choosing which individuals breed and selecting particular traits. So we were becoming an evolutionary force, less subject to, the winds of bad weather and not being able to find food. We started farming because it was, we didn't like the unpredictability of going out there and not being able to find something. The very earliest evidence of animal husbandry is from the area around the Fertile Crescent, somewhere between 10 and 12,000 years ago. And what probably happened initially, what archaeologists believe happened based on the zooarchaeological record, which is animal remains found at archaeological sites, is that people decided that driving something to extinction was not the inevitable consequence of keeping their families fed. But if they could learn something about the breeding strategies and the natural history of the species that they were relying on for food and change their hunting strategies a bit, then they could maintain, even support, even grow these populations. So you start seeing strategies in this zooarchaeological record of people only taking males, because if you leave the females, then the population can continue to reproduce. It might even attract males from a different population. So you get new individuals in the population that you're relying on, or only taking older individuals. And again, a strategy to sustain this population. And then close on the heels of changing hunting strategies comes corralling individuals and actually keeping them close to your settlements so that you don't have to go out and try to find them. And then you really see the changes happening where the individuals who are scared run away. And so they don't get brought into this next generation. And the individuals who are willing to take cues from people stick around and become things like cattle instead of aurochs, or our domestic sheep and goats and pigs and chickens and things like that. So, we've been manipulating things by taking advantage of how evolution works without understanding it for a long time. And now that we understand it, we can intensify our manipulation of other lineages and perhaps even learn to meddle better than we have been in the past.
If you just take dogs, maybe 10,000 years, that we've been changing them, there's such incredible genetic variation. Well, I don't even know how big the percentage is, but at least the way they look, there's huge variation in the way they look and some of their traits over 10,000 years. Do you think that when we think about millions of years, evolutionary forces that are at work amongst things, are we in a place where we can start to understand this complexity, or is it still something that we're just learning about every day?
About evolutionary complexity? I think this is probably something where we're still learning about, we're still learning to appreciate. It hasn't been that long that we've been able to sequence genomes from lots of things. And we're still in the process of generating genomic data from the diversity of life on Earth. And I think we continue to be surprised. One of the things that has been surprising to us, but in retrospect shouldn't have been, is that the more genomes we sequence, the more we realize that things that we consider to be different species actually continue to exchange genes for a long time, like brown bears and polar bears. Every time their populations overlap, they exchange genes, and dogs and wolves continue to exchange genes. And it's the norm that if something is not unable to exchange genes, humans and Neanderthals, for example, they do. And this is just one example of the complexity of the way biodiversity evolves and is sustained and is changed by things like climate changes or a new predator like people on the landscape that we didn't really appreciate before. And I think we are, and also just how genomes work. We're still really early in trying to understand this. One example of that is, there's a really cool project going on right now. It's led by Revive and Restore, which is a nonprofit out of Sausalito. And they are trying to come up with a way to help the black-footed ferret, which is a little carnivore that lives across most of the mid plains of North America to survive.
They have a mite or something, right? That's infected them.
No, it's plague. They get plague. Yeah. And they eat prairie dogs. The prairie dogs have been infected by plague. Plague was introduced from Europe. And so now all the prairie dogs have plague. But the black-footed ferrets were one of the original species listed with the original Endangered Species Act. And they were brought into captivity. They tried captively breeding them. It didn't work. That population went extinct, that they were thought to have been gone. And then a population was discovered near Meeteetse, Wyoming. And so everybody had a second chance, brought these individuals into captive breeding. The population in the wild seemed healthy for a bit, but that didn't last very long. They started also getting sick, combination of plague and distemper. So they brought all of the remaining individuals into the captive breeding facility. And it's great. They can make loads of captive bred black-footed ferrets. They could release them into the wild. And I think there are about 500 or something like this black-footed ferrets that are alive in the wild right now. But they still get the plague. So they eat a prairie dog, they get plague and they die. You could vaccinate them, but that's not a particularly sustainable conservation strategy. So Revive and Restore partnered with the San Diego Frozen Zoo and the Smithsonian Institution and a couple of other organizations to see whether they could turn to new biotechnologies to help save the black-footed ferret. So they had two strategies running simultaneously. The first one, which was super successful, was that San Diego Frozen Zoo had individuals in their collection that were from the first attempt at captive breeding. So their genetic background is different from the individuals that were captured the second time. And a year ago, they were able to clone one of these individuals using tissue that's about 30 years old. And this individual, Elizabeth Ann, was born about a year ago, and she will be introduced into the captive breeding environment. She will be the 8th founder lineage of that environment. And her genetic diversity is going to be the only diversity that comes from a different population than the other 7. So that will be a welcome burst of diversity into this population. But it probably isn't going to solve the problem of plague. For that, we have to turn to similar technologies that George Church and the company Colossal are using for mammoths. And that is the domestic ferret evolved along with plague in Europe and is naturally immune. And so if they could identify what about the DNA of domestic ferrets makes it immune to plague, they could then transfer that using gene editing to these captively bred black-footed ferrets, creating a black-footed ferret that is mostly black-footed ferret in its DNA, but a little bit domestic ferret, and that allows it to survive in its habitat. Problem, I have no idea. Have some good ideas about where to look, but no idea what parts of the genome will make it immune to plague. But it's still an exciting way that you can imagine trying to understand the complexity of the way that genes are interacting with each other and the way that genomes work, and our new biotechnologies to help us to help other species adapt to changes to their habitat that we caused, that evolution is not going to do this. There's too few of them, it's too fast. There's no way that natural selection is going to save these species. So if they're going to survive, it's going to be because we do something. And this is something that we might eventually be able to do.
How would you isolate a resistance to plague? You're looking for how many genes out of the billion genes?
Don't know. They don't know. So they're looking specifically in regions of the genome that we know from other studies are associated with disease resistance. And so they're doing trials, not with animals, but with cell cultures and dishes and labs, and challenging different edited cells with plague to see whether there's a response. So there's no.
And are they trying synthetic biology to do that, or are they just trying the genes, swapping the genes?
Well, you need synthetic biology to swap the genes, but yeah, not making new ones yet, still looking specifically at genes that evolved and genes that we know work somehow, but we don't understand how, to see if we can better understand that system. It sounds crazy, but there are actually examples where this has worked. There's one example in particular that I love, which is the American chestnut tree. Do you know the story of the American chestnut tree?
It didn't die out in the eastern forests, and then we had to try and figure out how to bring it back? Or tell us the story.
Yeah, so this tree was, there were billions of them in the eastern forests until the early 20th century, when a fungus that was introduced from Asia started to infect them. And the fungus gets into the tree and it releases an acid and it burns holes through it that the fungus then uses to tunnel through the tree. And within a couple of decades, all of the trees were dead. They succumbed to this chestnut blight. They're not entirely dead though. This zombie trees live as roots underground, and every now and then they'll shoot up these little shoots that survive, rarely long enough to fruit, but until they are also killed by the fungus. There are also these pockets of, I call them expat trees. They're trees that people planted as they were moving from the east to the west in the 17th, 18th, 19th centuries, or 18th, 19th centuries. And there are some stands there, but there's chestnut blight in most of these stands. But there's a team from SUNY in New York, led by Bill Powell, who decided, and the American Chestnut Foundation, that they would really like to come up with a way to save these. And so they started by hybridizing Asian trees with American trees. And this worked to some extent. They had some bit of resistance, but they also just weren't very good at living in North American forests because they hadn't evolved in these North American forests. And the more Asian genetic background they had, the more resistant they were, but the worse they were at thriving in these natural habitats. And so they were looking for another solution. What they discovered was that, lots of plants deal with fungi that do the same thing. And they've evolved ways of squelching that acid that's burning the holes through them and killing the trees. And so they went through lots of different plants looking for genes that have evolved to deal with this type of fungus. And they found one in wheat. They could have picked anything, but they picked wheat because wheat's very well understood. It's easy to grow, whatever. They isolated this gene that makes a protein that neutralizes the acid that the fungus produces. And they inserted it into the American chestnut tree genome. And so now they have this strain of trees called darling trees that is mostly American chestnut trees. It has this one gene from wheat. And that gene makes a protein that neutralizes the acid and allows the tree to live with the fungus. And the fungus doesn't kill the tree. And so right now they're jumping through all of these hoops for regulatory approval for a tree that is intended to restore forests. And maybe we can eat these American chestnut trees. And hopefully this is real progress. I mean, this is an example where they did know what gene to look for, and they were able to move just this one gene into this genome that they can now backbreed with all of the expat trees and all of the little root trees that are there, and reconstruct a ton of diversity that was in the American chestnut trees, but with this one little gene from wheat.
That's a point, though, the audience needs to understand. The last little point that slipped in there, and maybe we can eat these. I mean, I think for people who don't understand the enormous repercussions and consequences of the work that you do that are really extraordinary, how do we tie that into the world that we live in? And not everybody has this sort of science mind or really understands. That's a huge.
There was a comment period last year because as part of this approval process for the chestnut trees, and one of the people who commented his name is Jim Sneed, and he is an elder with the Cherokee, and he commented, speaking for himself, not for everyone, that he was looking forward to a time where we could restore this system to which he and his people and his culture had a long interaction with, and he was excited about the possibility of having this tree back that meant so much to him and to his culture, and the chestnuts that meant so much to him and to his culture, and excited that science might be able to bring some of this back that was what was destroyed. And I think a lot of us think like this. I mean, I love the idea of having American chestnut trees back. I don't care if it has a little gene from wheat. It doesn't bother me. I mean, I eat wheat anyway, so I'm not worried about eating a gene from wheat that I already eat just because it's in a chestnut tree. Right? So I don't think there's any danger in that. There's some strangeness involved with the idea that we've taken a gene from wheat and put it into American chestnut tree. I'm not going to deny that. And certainly people deserve to be able to make a choice about whether they want to eat that kind of thing. But I don't see it as risky. And I do see an enormous benefit to the forests that will now have this foundational tree back, and to the animals that lived in these trees and relied on these trees and ate the nuts from these trees that will have this source of food back. It's part of building back this habitat that existed before we did away with it.
How important is it for us to build these seed banks around the world that protect the genetic diversity that we don't actually know what it might be useful for? You know, there's no clear indicator of what it might mean or not mean.
Hugely important, and not just seed banks, but frozen tissue collections like in the San Diego Zoo. If people hadn't thought, 30 years ago, let's cryopreserve some of these tissues because they might be useful in future, we wouldn't have Elizabeth Ann, and we wouldn't have Kurt, the Przeworski's horse that was born recently. That will also inject a whole bunch of new diversity into an endangered horse population.
Where is that one in the steppes in Central Asia?
Yeah, that's where the horses live. But Kurt lives in Texas.
Oh. maybe not getting the same sort of vibe in Texas that he would in the steppes.
Last I heard that they'd introduced a girl for him to play with. He's not old enough to breed yet, but they're trying to.
Yeah. Wow. I have a question. In the deep sea, we have fish that have developed an eye this big, right? And we have eyes and birds have eyes. So that's a sensor to read light. Are there universal principles to life? Like, are they always searching for certain things and then developing tactics to deal with those things that we have here on Earth?
It's an interesting question, kind of phrased in a very not evolutionary way. We're not searching. Evolution doesn't search for something and then get it. Evolution is chance. Some mutation happens and it causes some phenotypic change, some physical change, and the individuals that have that physical change somehow do better than individuals without it in the next generation, and that's how it works. And we do all, we all share a common ancestor. Everything that's alive today goes back to something that was alive. Luca, the last universal common ancestor, many years ago, lots of years ago. And so there definitely is the same starting material that we've worked with, but there are some things that may be similar pathways or trajectories evolved multiple times. I know that you should get somebody who's really interested in sort of early origins of life and deep phylogeny to come and talk about that stuff. It'd be fascinating. I would definitely tune into that episode and learn more about it. It's kind of outside my expertise to talk about.
Beth, thank you. I mean, this has been incredibly interesting and challenging for my brain that is more Neanderthal probably to understand and comprehend.
Well, you could get yourself tested. You could have a competition to see which of you has more Neanderthal.
Well, I have a terrible 23andMe story where they said I had no DNA. So there you go. Well, we can talk about that another day. But
I can reliably say as a scientist that that's not true.
Okay. Well, I didn't know how to do it properly. But anyway, this was really wonderful. And for all of you watching, listening, read Beth's books. Okay. Read her books. Find out more about what she's doing. And
Yeah.
it's
Thank you.
wonderful work. And it's
It's been
really
fun.
it's so
Thanks
a lot.
lively and fun to talk about it. Thank you
Yeah.
so much.
Thank you for sharing with us.
Thank you.
So many interesting topics. Thank you very much.